Today, we are taking a deep dive into the world of medical device reprocessing and the tools, technology, and systems used to transform how the cleaning industry approaches hygiene, safety, and sustainability. Joining us today are leading experts in our industry at the forefront of innovation, helping healthcare facilities clean more effectively and safely. And as you can see, we have three guests with us. They're going to introduce themselves when they start to speak, but it's great to have John Howell, John Harp, and Brandon Byer with us today. Our topic is an important one, and John Howell is going to start us off with our first question. So, John, are you ready?
I am ready.
All right. This is a long one, so prepare yourself. We're going to start with foundations and safety. What is medical device reprocessing, MDR? Why is it critical to patient safety? And how do healthcare associated infections, or HAIs, connect to effective or ineffective MDR? Hope you can handle that one, John.
First of all, thank you for having us, Jeff. I'm John Howell. I'm Global Business Development Manager at Novonesis for medical device reprocessing. I basically help customers around the globe utilize enzyme technology in their medical detergents. So, to answer your question, MDR or medical device reprocessing is essentially when you go into a healthcare facility and you have a procedure, whether it's surgical or diagnostic, you have to have our instruments are used on you that have been used on other patients, and they're going to be used on subsequent patients. So, medical device reprocessing is really what happens to that device between use on you and a subsequent patient. How is it cleaned? How is it processed? And how is it made safe for subsequent patients? But you can imagine those instruments are contaminated with blood, mucus, bodily fluids, tissue, and microorganisms. All those things come from you. You don't want that on the next patient. So it's really about patient safety. How do you ensure that that process is the most adequate and the most efficient process so you take care of the patients that get subsequently are going to have those instruments used on them? And one of the biggest concerns for patients is what we call the healthcare acquired infection, or an HAI. Healthcare acquired infections are infections that are developed and obtained when a patient is in a healthcare facility, whether it's a hospital or an ambulatory service center. The biggest problem is once you get these infections, in some cases, we get infections when we're at home and they're fine. You can use an antibiotic over the counter or a prescription antibiotic, or you can even use a topical cream. But in some cases in healthcare facilities, they can be resistant to a lot of those things. And if somebody were to acquire one of those, it could be a lifetime or days, weeks, months, and even years battling through these infections. It's a financial toll, it's a mental toll, it's an emotional toll on the patients and everybody involved. So it's absolutely critical that when you are in a healthcare facility, and these HAIs, let's be honest, they can come from anywhere. They can come from the air, they can come from surfaces, they can come from textiles, but they can also come from an inadequate medical device reprocessing. So it's really important that in order for patient safety, that medical device reprocessing is at the utmost efficient and protects the patients in the end.
I'd like to ask you this about standards and guidance, because there are entities out there to consider. What do the CDC, the FDA, and WHO emphasize about the role of cleaning with MDR, including validation, documentation, and risk management?
Yeah, so great question. And I think one of the first things to understand is medical devices themselves or medical detergents themselves are not regulated. They are class one medical devices, which is essentially the same as a Band-Aid. All right. So when we talk about these bodies like the CDC, FDA, World Health Organization, when they're talking about rules and guidance and standards, they're really talking to the medical device manufacturers, the ones that are actually making the devices that are used, right? So they give them, strong guidance and standards on. They have to show how that the device they're manufacturing can be cleaned, that it can be sterilized or disinfected depending on what's being cleaned, and that they can validate that. And then these device manufacturers, they have to take that information that they develop and they have to give that to healthcare facilities and hospitals and tell them exactly how to clean their devices and how to get the best outcomes out of their cleaning and disinfection and sterilization, whatever it may be. Now, detergent manufacturers, on the other hand, they don't have these types of guidelines. So a lot of times they will utilize what the device manufacturers are kind of put through. So they kind of follow the guidance that the CDC and the FDA give to them. And they kind of just follow along with that and say, okay, this is how we have to prove those out. And once they develop theirs, they have to have developed their own IFU or instruction for use. Once they do that, hospitals should have to follow that. Healthcare facilities should have to follow that IFU. They should have to follow the guidance from the device manufacturers. Now, what's important here is all of these, the FDA, the CDC, World Health Organization, one thing they will all say when it comes to medical device reprocessing, cleaning is absolutely a critical step. If you don't clean properly, you can't properly disinfect or sterilize an instrument. So they put a lot of emphasis and onus on how do you clean these instruments. And medical detergent manufacturers, that's where they're focused in. How do we clean these to get the best outcome? And so once all that is provided to healthcare facilities then have to take those and turn those into protocols, established protocols, and they have to ensure that the staff is adhering to those protocols that they have written. And then on top of that is the Joint Commission. The Joint Commission is kind of like a survey or an auditing body that comes in through healthcare facilities, and they want to know, are you doing the right things? Are you adhering to the guidance that are put in your established protocols? So they can go into a reprocessing facility and they can look at it and say, where's your documentation? Are you using your equipment in the right manner? Is your equipment up to standard? So there's a lot of oversight to ensure one thing, and that's patient safety at the end of the day.
I want to talk about day-to-day practice and challenges. So, how does MDR work in healthcare facilities from the point of use through cleaning, disinfection, sterilization, inspection, packaging, and then release? Can you talk about that?
Yeah, absolutely. And there are two kind of flows. We have traditional instruments, which might be your stainless steel instruments, your spreaders, your plastic instruments like trocars, retractors, and then you have endoscopes. So just start talking about like the traditional instruments, so to speak. Once they are used after a patient, they're typically treated with a pre-treatment foam or a spray or a gel, some kind of a foam or gel. And that's meant to keep the surface wet until they can be reprocessed. Once that's done at the point of use at the patient bedside. They are sealed up and transported to a department where they will be further cleaned. Once in the department, they will go through sorting. They'll go through a potential rinse and maybe some scrubbing or brushing to remove any residual material. And after that, they will go into a soak in a sink, which has an enzymatic detergent or some kind of detergent in that sink. They'll soak in there for maybe 2 to 5 minutes, whatever it may be by the established protocols. They will come out of there, and then they will go to a ultrasonic in many cases, not always, but an ultrasonic bath, also with a detergent in there to help clean further. And then the final step is usually to a high-powered washer disinfector, which is like a fancy dishwasher, but with a lot more horsepower to it for medical cleaning. Once it comes out of there, they can go through inspection to make sure that there's no residual debris, no damage to the instruments. Once that's done, they'll be sorted again and put into surgical kits. They'll be wrapped and they'll put into a high steam sterilization or an autoclave. And then they'll be taken out on the other side once that's complete and put back into circulation for the next patient. Now, if you're talking about endoscopes are used at the patient bedside. They're done. They get wiped down with potentially a detergent type wipe. They will also go through and aspirate detergent through the lumen to remove loose debris at the bedside. Again, they'll be sealed up and transported to a reprocessing center. And then once they're at the reprocessing center, they go through a leak test to make sure that they pressurize it, make sure there's nothing leaking and there's no potential for water to get in and damage anything. And then they will take it through a manual soak, which is a detergent flowing through the system, having a brush in there to clean out the lumen. Then it will go to high-level disinfection. They cannot be steam sterilized, but they go to high-level disinfection. And eventually it will go on to a drying stage to remove any water to prevent any kind of films or organic biofilms from building up. And then after thorough drying, they go to storage and they're ready again for the next patient. The challenge here is just the amount of these instruments that come into a facility and the complexity and all the different things you have to clean. So it can be quite a daunting task.
I hate to ask the question, do you see this happening in facilities as it should be?
You know, I've been in a few facilities and it does happen correctly, but I'm not aware of all the established protocols that particular facility works with. But I do see that general flow going. How well it's working at the end, I don't see that as much. And of course, they don't want you to always see what's going on. But I will say this, healthcare facilities do put a tremendous effort into making sure that this is done properly. Technicians are trained, they have to go through accreditation to make sure that they are capable of cleaning all these things. But from the a few that I've been in, it's typically done in the right way. Now, is it adequate? I don't see that part, but overall, people are doing the right things.
Yeah, it's a challenge, but the payoff is avoiding these infections and keeping people healthy. So,
Absolutely.
yeah, good process you described there. Our next point, we're going to talk to John Harp and then Brandon Beyer on this one. It's about the cleaning professional angle here. And John, we'll start with you, and be sure to introduce yourself once I give you this long question. For the for those new to the topic, let's talk about enzymes. What are enzymes, and how do they differ from traditional chemical cleaners, and why are they especially valuable in healthcare and for medical devices? Tell us about that.
So I'm John Harp. I'm supporting on the technical service side within our medical device reprocessing team, also on the global stage supporting John Howell, and happy to be here to address some of these really critical questions related to medical device reprocessing. And so at Novonesis, we are extremely proud of providing biosolutions to the globe. And biosolutions are things like enzymes, things like microbes, things that come from nature, which can be used for a number of different types of products and a number of different types of consumable items that people use on a regular basis. And so in medical device reprocessing, we're extremely excited about enzymes. And enzymes are these little tiny proteins, that is, these chains of amino acids that are folded in very specific ways, and they act as catalysts to make a change on a particular substrate. And so this folded molecule creates a pocket that then creates an active site. And so it's that active site that then this action occurs. And so with these biological catalysts, which are found in just about every living organism on this planet, we are excited to use these, find the right ones that can then work on the right types of soils. So in medical device reprocessing, of course, we're talking about soils in blood and tissue, fats, starches, and other complex soils that could then be found in and on reusable devices. And so by being able to specifically target these soil types, you can then ultimately get back to what John mentioned, which is that you cannot disinfect or sterilize a surface if it's not cleaned well. So by using enzymes, you're able to target these soils to remove them in an efficient way. And so I'll let Brandon talk a little bit more about the chemical cleaning process and cleaning in general. But what we do know is that enzymatic detergents are very good at removing organic soils. And they're also safer to handle as it relates to alkaline detergents, which would require some extra precautions, extra things to consider during that cleaning process. And we know that enzymes can work very synergistically alongside surfactants to make sure that you're cleaning that surface as well as you can. So that way you can then reduce some of the manual scrubbing that might be needed to efficiently clean, as well as ultimately maintaining good equipment life, because you're not having to sort of wear down that equipment using harsh chemistry. So enzymes are very dynamic, they're very specialized, and they're very powerful for medical device reprocessing.
Well, thank you, John. Appreciate that. Brandon, he has tossed the baton your way. So, same question to you. Introduce yourself and then tell us what you know about this from the cleaning angle.
Thanks, Jeff. So, Brandon Beyer, I'm a technical manager within Solutions. I'm kind of the odd man out in this dialogue. So, you've got John and John, two technical scientists with Novonesis, and Novonesis is a producer, right? Solutions is a chemical distributor. So, the question is, why am I here? We have a global partnership with Novonesis, in particular in this space. And really, what Univar Solutions represents in this space is kind of a one stop shop for cleaning solutions. Yes, we are a predominantly logistics company, but we have a whole series of labs, technical professionals like myself, and the contact to people like John and John. And we have access to the enzymes and the other parts of the cleaning formulation. So that's really the spot my team sits within is formulation. It's bringing all these things together to bring a fully formulated chemical cleaning product to, say, for this place, for this situation, medical device reprocessing. So really what we're trying to solve at any given time, and just kind of getting to the baseline, we're talking about cleaning. It's the basics. The art of cleaning is temperature, time, mechanical action, and chemistry. And at that core is water. And John Harp had just mentioned, like what can enzymes really do in this space? It's simplify that wheel. It can reduce time. Enzymes are catalysts. So one enzyme, in theory, if it didn't denature, could just go through and turn through biological soils. It can reduce temperature. Enzymes come from physiological areas, right? So let's say you have a soaking bath that comes in about 104 Fahrenheit, 40 C. Maybe it cools off over time. The enzyme doesn't care. It will keep working. Whereas some of the more traditional cleaning agents, surfactants, chelants, polymers, do have a little more kinetic or thermodynamic requirements where the enzyme can just keep doing its work. So by the time you get to the point where you're either scrubbing or rinsing, it takes less scrubbing, it's working easier, potentially use less surfactant in the space. And I think the most important thing, surfactants are designed by chemists in a lab. Enzymes really are designed by nature for exactly these soils, proteins, fats, sugars, carbohydrates, whatnot. So they have a real special place within the cleaning wheel for medical device reprocessing.
Our next question and point is for you, John Harp. It's about innovation and next steps. So, how does Novonesis approach enzyme research and development to ensure safety, efficacy, and stability?
Like I said before, the enzymes are natural. They come from nature. And we have some very cool videos at Novonesis where we're seen walking through the woods and looking for very specific, fungal species and other life forms that exist in nature that are breaking down various soils. And this is where we go for the source of our enzymes. Of course, once we sort of start to isolate some of the key, what are known as genes, which then produce the enzymes, we do some really complex and really excellent work in the lab to understand those genes, to understand the output of the enzyme, to make sure that the enzyme is as stable as possible where it needs to function and in the environment that it needs to function. And so there's a significant amount of laboratory work and time that's spent in finding the perfect enzyme for the right application. And this is no different when it comes to the medical device reprocessing. We've looked at a lot of different soil types to see how those enzymes can work in those particular applications. And so the unfortunate bit around efficacy testing is that there's currently no universal standard for testing enzymatic detergents. And there are, quite a few standards that can be used, or I should say internal methods and indicators that can be used, such as TOSI and STF, GKE, ChemDye, et cetera, that can be used to showcase the power of enzymes. And this is the language that we still like to speak when we talk to those interested in medical device reprocessing with enzymes. But we have a little bit ways to go as it relates to the universal standards. And we really want to make that work. We're currently using and involved with ASTM and various other ISO methods to make sure that these frameworks are being explored in the right way when it comes to enzymes and medical device reprocessing. And so we really need to take all those sort of elements into consideration and do it in the right way, because enzymes are very powerful. They have to just be used in the right way and shown those benefits in the right way. And so when it comes to formulation, innovation, and performance validation, one of the things that we're very proud of is the ability to blend our enzymes in blends that actually offer value for relevant soils in these applications. So it's not always just about going after proteins, because sometimes those proteins could be in a mixture of other soil types, such as complex organic soils that might be in the lumens of these highly technical devices. And so by going after the elements within that soil, you can then really break the soil down to then have good disinfection. And so going after validation claims, making sure that you can showcase the efficacy within good time frames, because you also don't want to just say, oh, you're going to have to add 3 hours to your process. You want to sort of maintain within the right time frames. And then, of course, you want to be compatible with those medical instruments themselves. You want to work with the medical instrument and get to the proper clean. So yeah, that's sort of the round the horn sort of answer to that question.
I appreciate that. And in a former life, I ran a cleaning company, and we use a lot of enzymes in different applications, but they were fantastic, and it really solved issues that from odors to soils. So, even more important, today's topic on what they're being used for. John, how are you still awake? You still with us?
I'm still with you. Still
All right.
awake.
You get the wrap up here.
All
So,
right.
so what does the future look like for MDR, and how can those watching learn more about it?
Yeah, so for the future of MDR is really about it's like any process, it's going to evolve, right? I think you're going to see like we're getting an aging population that's expected to double by 2050, and aging populations require, people over 65 require more procedures and more surgeries. You're going to have more surgeries as it becomes more and procedures as it becomes more available to people, and as medical guidelines change and they lower, like ages for colonoscopies or whatever. And so you're going to get a bigger volume, and you're going to see sustainability take more of a hold. And you're going to see device complexity as device manufacturers start developing into like the robotics space, which they're already doing. I think you're going to see a lot of changes. You're going to see a lot of changes in the medical device reprocessing, and device manufacturers, detergent manufacturers, and all the governing bodies are all going to have to kind of come together and continue to stay ahead of that. And I think for medical detergent manufacturers, one of the big things that they're going to have to do is they're going to have to stay ahead of and potentially reformulate and optimize their detergents to continue on with some of the device complexity, and how do they stay ahead of the game? Because at the end of the day, all this is about patient safety and how do we mitigate these healthcare acquired infections. You can reach us at novonesis.com or univarsolutions.com. And you can also reach John, Brandon, and I at LinkedIn. If you want to find out more and you want to ask some personalized questions, feel free to reach out to us. But there's a wealth of information out there, and we look forward to speaking with anyone.